It's just, it can't be done.
And it even comes down, it does come down to,
It's like just collecting cats.
Yeah, yeah, yeah.
that neurology
Yeah, yeah,
is an underfunded discipline.
yeah, yeah,
And it's huge debt.
yeah.
Everybody who goes through neurology,
Yeah.
so they don't get doctors.
Yeah. No, exactly.
They
Yeah.
don't get residents coming
Right. No, exactly. Yeah, no, and these you've got all, it's just all kinds of problems, because now at least most of these neurology departments now, part of their funding is just clinical work. And so you've got people that are bringing money into the department just because they're an attending at the hospital, and part of the money that comes to them goes to the department. And then you've got another group that have nothing to do with clinical work. They're simply researchers, and all their money just comes from overhead on their. It's just like these two departments should not be together. I mean, they should not be in one. Yeah. Oh, it's beyond.
and they can't get more than one partner.
Yeah. No, it's I don't. I mean, but these are what your points is. What are the solutions? Well, that's the only one ever. Blow it up and let's start all over. I mean, it's just,
And I would like to spend just 2 minutes talking to you about my two favorite professional philosophy friends in the world, and that is John and Bob, which is one reason I'm so excited that I get to introduce John and Bob Palooza. Because I've known them virtually my entire professional career. So I first met Bob almost exactly 30 years ago. It's like 2 months shy of 30 years, when I was a baby assistant professor and put on a conference at Mountain Lake Resort in the wilds of Virginia. And he showed up. And I read his stuff, of course, because, you know, that Helen Richardson, right? It's all over my dissertation. And it never occurred to me that, like, anyone except maybe my mom would come to my first philosophy conference. But he showed up with bells on his heels, and it's been great. It was great. And then then I came here for the SPP, whenever that was, 5, 6 years later.
Yeah.
Maybe
2000, maybe
Yeah.
2001.
I remember
Yeah.
Quinn was 3. That's how I remember it. And it was wonderful, except I couldn't find parking, which again was like the same issue tonight. I was like having deja vu all over again. So I was running in late, and I had to drag 3 kids with me, and they were all very small. But they were trying to be good, but they were mad at me because I was making them run, which they didn't want to do because of their clothes. And my son Quinn, he was about 2.5, maybe 3, and he was trying to be very patient, and he was coloring. And then I finished this godly long talk from his perspective, and then, God forbid, people started asking questions. And finally couldn't take it anymore. So he stands up with his little diapered self and marches down the center aisle with his arm raised, because you have to do that. Hold on. And somebody said, yes, I believe he has a question. And he says, yes. Are you going to be talking all day, or give someone else a chance? I said that was the last question I was going to take. Then I came here for my sabbatical, which was shortly thereafter, and Bob's daughters helped raise my daughters, which was a very nice connection. And then I came here later as dean, and Bob was still here. And so it's been fabulous. Our lives have just crisscrossed throughout my whole professional career. And he's just been a wonderful supporter and always cheerful and willing to talk about pretty much anything and everything. John, I have known even longer. I first met him in graduate school. And one of the things that I've always appreciated about my relationship with John is that we almost agree on everything. But it's like that last 2%. We are just like, no. And so we have had some of the best, from my perspective,
Mine too.
conversations, and sometimes arguments, depending on like how much alcohol has been consumed, regarding some very deep and important distinctions in the philosophy of science. So I'm just super excited to be able to be here and introduce John at this event because it's very meaningful to me, and I am the center of my own universe. So without further ado, let me introduce John, who is going to be talking about neuroepigenetic explanations, neither mechanism, trademark, nor pathways, trademark, but a smidgen of both.
Right, exactly.
Thank you.
Thanks so much, Valerie. And yeah, thanks, Valerie. And it goes back a long way. And thanks, Bob. And I've got a great story to tell about Bob, but I'm going to save it for the toast session, because it was an early lesson from me on how people that are really well known should treat younger people who are trying to get into the profession. And I've tried to carry on the lesson that Bob, and I'll talk about it tomorrow, the lesson that Bob taught me at a very young age in my career, and I've tried to carry it on from on. And so let me get started today. And I know, and a lot of you know, I have huge visual problems. I'm going to try to present this talk from here, but I may have to revert to the screen for some of the wording here. But first, let me start by introducing these two puzzling terms in my title: MechanismTM and PathwaysTM. Trademark. So now I think we all know what MechanismTM is because we've been dealing with it explicitly since 2000, Mackiner, Darden, Craver, and of course all of Bill's work that was at the same time developing this idea specifically of what mechanisms are in the life sciences. Now, pathways is probably a little less known to you, but if you've been keeping up the literature, that's Lauren Ross's recent work, which she proposes as a challenge to the ubiquity of MechanismTM in causal explanation in the life sciences. Now, for a couple of preliminaries, for this event, I really wanted to talk about something that related directly to Bob's work. So, Bob and I were department colleagues from 2000 to 2009. Bob, of course, is hugely known for his work in the philosophy of biology, especially evolutionary biology and evolutionary psychology. And of course, as we've seen, and we'll see it every talk here, the 1990 book, widely acknowledged as one of the foundational documents of new mechanism in the philosophy of science. And I'm definitely going to have to revert to behind this so I can better read these particular things. So I thought, why not talk about neuroepigenetics? Now, that's a new hot addition to biology and to neuroscience. And so it combines the kind of basic idea of epigenetics, the study of changes into organisms caused by modifications of gene expression rather than alterations of the genetic code itself. But it also applies this directly to the study of how epigenetic changes in gene expression affect the nervous system. And so it was a way of combining sort of this general interest that both Bob and I have had for a long time. It also came about because a couple of years ago, and yeah, you're reading that time right, Antonella Tramaceri postdoced with me during the infamous fall of 2020. And the reason I hired her at the last of my postdoc money was because she knew neuroepigenetics and it's something that I wanted to get into. Antonella trained in the Parma lab, so she was part of the mirror neurons group, but she wanted to know more about the way that these particular neurons were formed. And so I had her postdoc with me so I could learn some neuroepigenetics. And the first result of this was this paper that just came out this year, Neuroepigenetics in Philosophical Focus, a critical analysis of the philosophy of mechanism, which came out in Biological Theory, or maybe it's coming out soon. Now, let's start with some science, and I mean real science. Okay. Now, I want to talk a little bit about the work of Mike Meany and Mosey Zerf, McGill University Faculty of Medicine, because back in 2004, this group published what is one of the most highly cited papers in this entire literature, Epigenetic Programming by Maternal Behavior, now cited over 7,000 times because it's a beautiful example of neuroepigenetics research. And I'll have to be brief on the science today, but let me kind of give you the background of the particular science in Weaver et al. 2004. Rat maternal behavior is highly stereotypic, and there's two particular measures of rat maternal behavior that this group focused on. Licking and grooming, the amount of time the mom spends licking and grooming the pups, and the particular kind of nursing style that the mom adapts. Now, the interesting thing about this stereotypic behavior in rodents, rats in particular, is that it's almost normally distributed. So you can take the mean on both these measures. You can go back one standard deviation below the mean and one standard deviation above the mean on both these measures and have your high and your low moms adopting this particular behavior. Now, the cool thing about this particular behavior is that the offspring of high LG-ABN moms have lower blood corticosteroid levels, and they show a more modest response in the typical kind of stress tests you use to measure rodent response. Now, so relaxed pups result from the high interaction dams, and fearful pups result from the low interaction dams. And the cool thing about this is these effects persist into offspring adulthood, whereas the maternal behavior only lasts the first 6 days postpartum. So here's the cool thing that Weaver and company did. And what they did is they went and did the molecular genetics involved in these particular two groups of offspring mice. And they were able to show this. You're looking at the sequence map of one particular exon in the promoter region of the hippocampus glucocorticoid receptor gene. And I'll be happy to explain to you why they chose that. They didn't just pick it out of a hat. And then including its various the sites where the methylation takes place and the binding site for node growth factor 1A. And what they were able to show by looking at the molecular genetics of these two groups of offspring is that you had increased methylation at these particular sites in the low interaction offspring. So the low interaction offspring had more methylation at these crucial sites than did the high interaction offspring. And why that matters is because the decreased expression of that receptor gene in these hippocampal neurons is in the offspring is basically in the low interaction moms leads to a decrease in the hippocampal sensitivity to the glucocorticoid negative feedback. And that produces an increased corticotropin releasing factor synthesis in the hypothalamus pituitary adrenal axis. And that leads to increased fear, stress response, persisting into adulthood, right? Because these epigenetic mechanisms are controlling for the glucoreceptor gene expression. Okay, so here's the basic picture now. You've got the relaxed offspring resulting from the relaxed moms, the fearful offspring from the fearful moms, and then what became recognized next was that if you did your cross-fostering studies, if you took an offspring from a high interactive mom and cross-fostered it immediately after birth to the low mom, and vice versa, took a low offspring and cross-fostered it to the high, well, the relaxed pup followed from the maternal behavior. So you could take one with the genetic background from a high mom, cross-foster it to a low to get the low treatment, and it behaved the rest of its life like a low. And similarly for the low, cross-fostered to the high. And here's where the molecular genetics came in. What this group discovered was that you got in the offspring, in this case right here, in the offspring that were high, that were cross-fostered to the low, you basically got the methylation pattern of the low, or offspring raised by the low. And on the high, the low raised by the high, you basically got the methylation pattern of the high raised by the high. And these changes began immediately the first day after birth when the mom started engaging in these particular behaviors. I mean, that is cool research, but it gets even better. What this group was able to do was they were able to do this incredibly cool molecular intervention experiment. The microapplication in vivo of trichostatin A, which is a histone acetylase inhibitor. So basically what it does is it interferes with the removal of the acetyl groups from DNA to increase gene expression and then also in the translation and the protein synthesis. And here's the results. So the trichostatin treated mice rescued acetylation at these particular sites, and with the predicted effects on decreased methylation at those particular sites in the trichostatin treated low mom animals, and the increased gene expression and the behavior, which was basically now tracking with the highs as opposed to the lows in your measures of stress, both physiological and behavioral stresses in the adult animal. Okay, now in terms of translational work with this, the Meany lab is into this kind of translational stuff, and what they were able to show was that using cadaver tissue, recent cadaver tissue from suicide completers with it, that's their term, suicide completers without early childhood exposure, with and without, and with some ingenious in vitro work with this dead tissue to basically make it talk, because there's a human analog in the human hippocampus genome for that particular homolog of the rodent exon. And what Pat McGowan was able to do was to show that you get epigenetic regulation of the glucocorticoid receptor in human brain associates with childhood abuse. And then later, and I won't try to pronounce this name because I'll totally botch the French, but a graduate student was able to show this for a more generally by exploring the genome. And I don't have time to talk about these details today, but Antonella and I talk about some of these details of the translational work in our paper. Okay, so here's the way that Weaver et al. In the 2004 paper frame their conclusion. And I really want you to pay attention to the language because it's going to matter for a point I'm going to make in just a little bit. Here's what they say. Our findings provide the first evidence that maternal behavior produces stable alterations of DNA methylation and chromosome structure, providing a mechanism for the long-term effects of maternal care on gene expression in the offspring. And I've outlined the particular word I want you to focus on. That is what they said. The maternal behavior produces stable alteration of DNA methylation. And I'm going to come back to that point in a little bit. But right now, I want to focus on that expression, providing a mechanism. Are they talking about a mechanism trademark in philosophy of science? And I think the answer to that question is no. Because first and foremost, this mechanism, you can see here, the mechanism of the high maternal interaction and the decreased methylation or the increased methylation is part of an entire causal pathway, basically, that runs from the maternal behavior through a number of steps in the molecular genetics, the gene expression, and the physiology. And it's that entire causal pathway that their particular story is embedded in. Furthermore, here's a diagram that Antonella did this really nice diagram for our paper about this pathway. And this is just simply does not seem to resemble a mechanism. The mechanism discovered by Weaver et al. Does not resemble the classic image that has come to be the representative image of MechanismTM. Furthermore, mechanists have really given little attention to the role of the environment in neural mechanisms. Bill is one of the few who has given sustained attention to the role of the environment. If you look at his 2009 paper where he talks about situating a mechanism in an environment and seeing how the system's behavior changes according to differing inputs from the external context. And that's the natural way for mechanists to handle environmental inputs, right? This is the natural mechanistic interpretation of the causal role of the environment. Environment is limited to generating a series of inputs or initial conditions on already existing internal mechanisms. But that is not the role that epigenetics is finding for environmental influences. These environmental causes, the moms licking and nursing behavior and the mom's arch-back nursing style, is literally building the gene expression machinery for the offspring's lifetime. So which cause of course, the lifespan physiological and the behavioral effects. So the mechanism is it's a part of this entire causal network that's generating the behavior, the mechanism that lasts a lifetime. And it's a causal component of this multi-step complex pathway. And this role is not so easily captured on any existing forms of MechanismTM. Now, you might ask, well, look, why not just extend the boundaries of the mechanism to include the environmental causes within it? Why not just go to extend the boundaries of the mechanism into the environment and then just kind of tell the mechanistic story there? Well, The problem with this has to do with the non-negotiable feature of MechanismTM. Nested hierarchies of mechanisms within mechanisms with all causes intra-level. Because that set of causes right there, the mom's tongue and the nursing posture, are just simply at a different level of mechanism than are the direct effects that Weaver et al. Was able to show on the molecular genetics of epigenetic mechanisms of gene expression. You recall Weaver's the thing I should have paid attention to. Our findings provide the first evidence that maternal behavior produces stable alterations of DNA methylation and chromatin structure and on. That is the story that they're telling. And this research tracking the different causal effects of these kinds of environmental variables, molecular genetic causal relations, just does not naturally fit into MechanismTM causal picture. Neuroepigenetic research, so to speak, on the hoof, neither assumes nor finds nested hierarchies of mechanisms within mechanisms. It finds direct environmental effects on gene expression machinery. That's how they talk. And if you're going to say, oh, well, they're presupposing this, you're putting words in their mouth. Okay. So in an interesting sense, I first noticed, this challenge is exactly the converse of my old ruthless reductionism challenge to MechanismTM. Ruthless reductionism insisted that molecular and cellular cognition is finding direct molecule to mind, well, behavior, causal relations. And this is the converse, right? Neuroepigenetics is finding direct macroscopic environmental to molecular genetic causal relations without any concern or recognition in their field of interlevel constitutive structure between the cause and the effect. Okay. Now, I also mentioned that Bill is one of the few mechanists who's paid any attention to the environment whatsoever. Okay. But in that same paper, that 2009 paper, here's something else that he says. Brains are active systems. So better to think of external conditions as perturbating the internal dynamics of its internal mechanism being modulated by external inputs. Now, that wording is certainly more on track with what neuroepigenetics are discovering and saying about their discoveries. Unfortunately, he doesn't say more about that in this particular paper, and there's still the assumption of the nested hierarchies of mechanisms within mechanisms and all causal relations being intra-level, mechanistic level. Okay, so that raises the question. Is what we were reporting in this sort of as the findings of neuroepigenetics, is this a better job for PathwaysTM, the kind of work that Lauren's been doing? And let me just say that this argument that I'm about to give was first suggested when Aaron Costco, who's back there somewhere, Aaron Costco and I were working on this in one of the philosophy and neuroscience at the Gulf talks, unfortunately the one where we had to do online because it was fall 2020. We sort of suggested this in our presentation. We haven't written it up yet, but I want to sort of let you show how while pathways may look like kind of an obvious way of thinking about what neuroepigenetics is finding, it's not so easy. Okay, so Lauren, of course, account of causal pathways in the life sciences, numerous publications, and of course her awarded NSF career grant that I'm happy to say I was one of the reviewers for it. So that was kind of cool. She got it. She gives numerous examples of this pathways concept in the life sciences. I won't go through them, but here's what she says about it. It refers to a sequence of causal steps that string together an upstream cause to a set of causal intermediaries to some downstream outcome. That's the pathways outcome. And given the way I sort of showed how this particular set of causal interactions work, from the mom's external behavior to the molecular genetics to the protein synthesis to the effects of that protein throughout the entire system, leading to the physiological and the behavioral outcomes, doesn't this sound very much like what Lauren is talking about? Well, here's some key features of Lauren's notions of pathways. Pathways involve a sequence of causal steps in some process. Check. Pathways track the flow of some entity or signal through a system. Pathways abstract from significant causal detail of a system or a process. Pathways emphasize the connection aspect of causal relations. No restrictions are placed on the components of pathways. You have the possibility of pathways working interlevel with higher and lower mechanistic levels. And finally, she thinks that pathways overcome philosophers' tendencies to think in terms of single causal relations to characterize the notion of cause in the life sciences. Okay, now that's what gives her her key differences between pathways and mechanisms. A single pathway, as she insisted, can be instantiated by different mechanisms. Distinct pathways can have a similar mechanism, and there's her example. And finally, pathways can be discovered with little knowledge of the mechanisms that underlie them. And he says, this is the case of many developmental pathways, such as stem cell differentiation pathways. Now, I want to focus on that key difference that Ross sees between her notions of pathways and the notion of mechanism. Because at first glance, pathways, TM, Ross, look very well suited for capturing neuroepigenetic science and practice, except for two of her key features. The first one is this one: track the flow of some entity or signal through the system. She gives a number of biological examples plus a non-biological example of flow, okay? But I ask you, in this particular pathway, what entity or signal is flowing through this set of causes and effects? I don't think that's such an easy question to answer because of the differences. There's not some kind of entity or even, I think, a signal that's flowing through, because to characterize the effect of that maternal activity and its effect at the molecular genetic level as the transfer of the signal of the mom's behavior, that seems to me like computational mumbo jumbo. This is just a direct causal effect. Now, here's the other one: abstract from significant causal detail. That's a key to Lauren's distinguishing pathways from mechanisms. Oops, there we go. But neuroepigenetic experimental designs exploit huge amounts of causal detail. I told you about the intervention experiment that's involved in the Weaver paper. A huge amount of molecular genetic detail was exploited to develop that intervention and to track that intervention through its effects. And that's where the causal detail was crucial to developing those experiments and drawing the conclusion based upon them. Okay, so there's the particular parts of the causal pathway that was discovered. And in this regard, neuroepigenetic research does seem more like what the mechanists would say, fleshing out a mechanism sketch rather than articulating a Ross-style pathway. So neuroepigenetics in actual scientific practice, in the way the experiments are designed and in the way that the experimenters conclude on the basis of their effects, seems neither mechanism TM full-blown nor pathways TM full-blown, but a smidgen of both aspects. Okay, now I've got a little bit of time still left, right, Val? Oh, okay. Well, I do want to say one more thing. I'll try to be brief with this because I really want to hear what people have to say. Is there a meta-scientific framework suitable for capturing this science? And I think there is, and you won't be surprised what I think it is. Right? This is the book that I wrote with Alcino Silva and Tony Landreth, Engineering the Next Revolution in Neuroscience, the New Science of Experiment Planning, back in 2024. There's Alcino right there, distinguished professor of neuroscience, neurobiology, and psychiatry at UCLA. And there's Tony. And of course, as some of you know, Tony's all in the family. He's my son-in-law. There's Kat, my middle daughter. And what we tried to provide in that paper was a meta-scientific study of some landmark publications from the first, at that time, 20 years of the science of molecular and cellular cognition. And early in the book, when we kind of choose the papers we're going to meta-scientifically analyze, we lay out what we call the three types of experiments that are dominant in molecular and cellular cognition. Identity experiments, by which neurobiological phenomena are discovered and their basic features are uncovered. Tool development experiments. This is a science that depends entirely on the research tools that the main labs have, and the big labs in MCC spend 80% of their time trying to develop new tools. And finally, connection experiments, the bread and butter of molecular and cellular cognition, where causal relationships are discovered between one neurobiological kind and another neurobiological kind. And since those kinds of experiments are so central to molecular and cellular cognition, we spend the rest of the 180 pages in the book analyzing them and the way that those results are integrated across experiments and across labs. By the convergent 3 analysis of the results from those 3 types of connection experiments, and by mediation analysis, where when a particular causal relationship between one neurobiological kind and another neurobiological phenomena have been established according to those measures, then you go try to find the intermediate causes that flesh out that causal story. Now, with one important addition added to what Silva Landreth and I did in the book, and that's another paper I wrote with Aaron and published a few years ago, Connection Experiments in Neurobiology, where we use the Weaver et al. Study as one of our key examples to derive this idea from. Okay, so now in this additional feature that Costco and I developed is the centrality of what we call multiple experiment research programs, with each component experiment designed so that its results can be integrated with those of the already established experiments. So the very design of these experiments pays attention to what has been discovered before, and the experiment is designed such that if it is successful, those results will be integratable into what we already know about this particular pathway, with the goal of finding evidence for multiple component causal pathways, for example, in molecular cellular cognition of learning and memory, like the cyclic AMP, protein kinase A, CREB pathway for late long-term potentiation, and declarative memory in mammals. Now, experiments integrated by design to elucidate causal pathways are not only prominent in molecular and cellular cognition of learning and memory, but they're also in these multiple component neuroepigenetic causal pathways, increasingly linking brain and behavior and environment with via real wet lab science, where the causal effects of the environment literally have effects at the level of the mechanism of new gene expression. Now, for a first stab at articulating the details of this picture, both the scientific and the meta-scientific, you can see mine and Costco's paper from 2018 and Tinella's in my paper that just came out. This was a first attempt to try to reflect to philosophers what a cool area of research neuroepigenetics is, what a great area of research it is, and how it can really lead to some interesting philosophical ideas, like the fact that there's mechanistic aspects involved in this research. There are pathways aspects involved in research. Neither side gets it exactly correctly, but it's a combination of a lot of the stuff that both, Carl and Lindley and Bob and Bill have been doing for 2 decades now, more than that, and the new stuff that Lauren's been raising with her investigation of causal pathways in the life sciences. So I'll stop for now. Look forward to having questions and discussion. And of course, let's all bear in mind that if there are no stupid questions, there are no stupid answers either. Thanks. Please, my vision wouldn't permit it.
I figured.
John, you are making a mistake.
Okay.
You need to reduce the maternal behavior to its mechanical molecular mechanism.
Tell that to the
Let me
neuroepigeneticists.
finish. Licking is nothing more than mechanical energy and heat energy. So you need to tell us the story of how that licking or those energies is being converted to chemical or electrical. It's not the maternal behavior.
Interestingly,
it's the stimulus of the energy. It's the input on the pups.
Yeah. Interestingly, you can use artificial means to do the licking and grooming and the maternal behavior, maternal posture, and it doesn't have the same effects.
Well, then you got to figure out what about.
Oh, yeah, I agree. I do. You're trying to articulate a causal pathway. Yeah, but look, the Weaver. Remember?
Say maternal behavior causes.
That's what the neuroepigeneticists are saying.
They're not philosophers of science.
Well, that's fine. But this is a philosopher of science who's trying to describe scientific practice. And there's scientific practice for you right there. Now, they are looking at these kinds of things, but so far, they haven't been conclusive. You cannot mimic the mother's tongue yet and get these effects. You cannot mimic the mother's nursing posture and get these effects. Now, the tools are pretty, you're just getting started, right? We could develop ones, and maybe we can flesh out by doing the kind of mediation studies that Silva and Landreth and I talk about in the book. And maybe we will tell a story, right? But right now, the conclusion that these guys drew, this group drew, was the maternal behavior directly caused the gene expression, the epigenetic mechanisms of gene expression. That's the title of their paper.
Oh, yeah, sure.
Yeah.
Well, a comment and then a
Great,
question.
sure.
I think it's easier to find an ecumenical stance between these two views because I don't think they're very different.
Okay.
That is, I think that mechanisms and pathways, I don't understand the distinction as you've drawn it or as Lauren has articulated it. So to me, it's pretty easy to find a middle ground there.
Okay.
But the question, you said something about levels in there, and I just want to make sure that you're right. You said there was no crossing of levels.
That you could. There's no account in this picture, and this is something Antonella kind of impressed on me. In the Silva Landreth Bickle picture, there's no strictures on where a neurobiological phenomena can fit into that particular story.
What's the stricture that you're imagining that levels supplies?
As I think we know that what we hear from the mechanists, all causation is intra-level.
No. That's what do you mean by level though?
Oh well, I'll bet yours level of mechanism. I have a different picture, but I'm just playing you guys' game right now.
What you just said is that a token cause doesn't causally influence the thing that it's part of. That's what you just said, and that's correct. But that has nothing to do with whether big things interact with small things. I mean, that's Wimsatt's point. Wimsatt's point a long time ago was tongues can influence molecules, can move muscles, can twitch neurons. Those aren't at different levels.
Well, I guess you and Bill
they aren't at different levels in the mechanistic sense. They're different levels in some intuitive sense of levels. That's correct. But the whole point of supplying a mechanistic account of levels was to say that intuitive sense of levels.
No, I agree. So I'm playing that. Now I have my own account, and Silva and I just published a paper last summer that basically says with these cool new research tools in molecular and cellular cognition, you can do your molecular interventions and measure your behavior just like we've done for a quarter century. But now with these cool mini scopes, you can basically get the mouse walking around with a 2-photon confocal microscope in its head, and we can image thousands of cells in as deep a structure as the hippocampus or even the hypothalamus. While the mouse has undergone the molecular manipulation, we can track the behavior. We can do all this now in a single experiment. So that suggests to us that when you get these cool new tools and you can do all this in one experiment, the whole notion of levels can just go out the door. I mean,
so two things are involved in the same experiment that can't be at different levels from one another.
Why?
Because we intervene on molecules to change the cells that they're parts of. to change the organs that
But-
they're parts of,
but we
to change the organisms that they're parts of.
That's just
and
talk.
That's a part-whole relationship.
That's just talk. We now can do it in a single experiment.
But I thought you were relying on the existence of tongues and
Yeah,
heads.
yeah.
and pituitary
Yeah, I
glands
do
and
now.
kidneys.
I do now because now I've got all the tools to do all of these interventions and measurements at the same time.
Yeah, I think maybe we're talking cross purposes on the notion of level. I'll just suggest.
Yeah, sure. Yeah, it may well be. But I mean, how do you and Bill solve, top-down causation without top-down causes? You restrict causation intra-level.
That's another paper.
Well, I mean, it is highly influential. Okay, it's a highly influential
The
paper.
paper says that it's a hybrid of a causal claim and a constituent and an identity claim. And so that's how you do it.
Well-
It's situated in a dialectic.
Yeah.
I mean, the goal of making top-down causation legitimate is that we can use it. We can, there's no problem because whatever your strictures are on causation, we can handle it.
But you do it by all causes being intra-level.
We're trying to make good interlevel
Yeah.
causation.
Yeah,
How could
yeah.
we be saying that then?
Well,
I mean, that's why,
isn't that
we've
the
been misunderstood for a very long time.
Okay.
Our goal was never to rule out interlevel causation. It was to take people who had particular conceptions of how causation had to work and to provide a framework in which one could understand top-down as well as bottom-up causation. And I mean, I think Carl was outlining.
But again, you don't do you have? I mean, here we go. Do you have higher level stuff causing lower level effects
Sure.
by just direct causal relation, just like what these people are claiming? Well, I mean, it means when you intervene this way, you can measure the effects at the mechanisms of gene expression.
That's not what we were worried about.
Well,
And we accept that.
Okay, well, I mean, but then what do you? I mean, that paper needs to be rewritten then because this is the standard. This is the yeah.
It's not situated. I mean, first of all, it's got a very restricted notion of leveling.
Yeah, I agree. Mechanism.
Super restricted.
Yeah, I agree.
Simply to, well, no, I would agree with the restricted notion because I don't know what a broader notion of level is for the very reason Carl just said. So the only notion of level I could make sense of is a part-whole relation. And then it does seem peculiar to people to say the part causes, the whole causes the part, or the part causes the whole.
Right.
Because they are the same.
Yeah.
The whole is nothing other than the parts. So we're giving you a framework to think about it. If that's the way you find that to be a legitimate concern, we can mitigate it.
Well,
and then we can talk about, as you say, if you mean by direct, simply I intervene here and detect here, fine. We don't want to sanction that.
Then that particular paper has been so widely misunderstood.
Yes, it has.
Okay, well, okay. Okay. Maybe then it could be rewritten in a way that would got that misunderstanding. But yeah, so you guys are fine with the higher level stuff, higher level mechanisms causing lower level stuff, and that's top down causation without top down causes. That sounds hard. I'm saying what the title of that paper is.
I think you're sliding all over the place with your notion of level and not realizing. That's my diagnosis of what's happening here.
Well,
is that in one context, you're using level to mean big things and little things,
Mhm.
things studied by one science and things studied by another science. And in another sense, you're meaning it in the top-down constitutive sense that we're using it.
Mhm.
And you have to recognize that our claims are bracketed by that very, as Bill was just saying, they're bracketed by a very particular sense of level. When you then move to this other sense, what I was calling earlier, the intuitive sense of level, that there are big, there's societies and organisms
Right.
and organs and cells.
Yep.
Like, why can't a society impact cells? Why can't planets, as we say
And
in
how
the
does that
paper-
causation
As
work?
we say in the paper,
Tell me how that. Tell me.
planets attract
Does that?
molecules,
But that causation work.
elephants
Give me. Tell me if I'm wrong.
smash
Tell me if I'm
leaves.
wrong. Tell me if I'm wrong. Tell me if I'm wrong. That causation works, however, by an intra-level cause at a mechanistic level, which is whereas the lower level stuff is a constitutive of that higher level cause. So the causation you're talking about really is intra-level. It's just the constitutive relationships that enable you to talk as though that's I'm just
That's correct.
characterizing the mechanistic view as I understand it.
Inter-level causal claims, just to repeat what's in
Yeah,
the paper, is
yeah.
supposed to be a hybrid.
Yeah,
of
yeah.
a unilevel causal
Yes,
claim
yes.
and
Of an intra-level causal claim with a constitutive relation between the higher level and the lower level.
Which is to say that the parts don't cause the wholes, rather the parts cause each other. and
Right,
the
right. That's what I'm
consequences
saying.
change the
That's exactly the picture I have here.
No.
Yes, it
Right,
is.
It's framed within, you've got a constitutive relationship. We're not worried about anything outside
Yeah,
of
yeah,
that constitutive
yeah.
relationship.
But
So your examples are outside of it.
no, but the cause is intra-level for you, for both of you. That's where the cause is. It just so happens that there's this constitutive relationship
You
that
start with constituent relationship and ask, can the whole cause the part or the part cause the whole? And
Right.
find that
And-
problematic. But we're
right.
inside the constitutive relationship.
right.
And then we ask, well, what we do
What-
want to say, if you affect the whole, it affects the parts.
Right.
How can we understand that?
Right.
Well, when you affect the whole, you change some set of parts.
Right. Because when you have a part affecting a whole, it's because the part affects the parts that are constitutive as a whole. That's the picture, right? The causation is intra-level. That's all I'm saying here. And you guys believe it. I mean, you guys.
Let's bring
Yeah,
in another voice.
Can I just make one point about that?
yeah,
If a flea bites someone
yeah,
and then they get sick.
Yeah.
A flea is not part of the somebody who got bitten and it's
Right,
small.
right.
It's intuitively at a different
Right.
level. And I think actually a lot of the cases that you're thinking about here with these sort of these epigenetic cases are something rather like that.
Mhm.
That it's going in different directions.
yeah,
Something big
yeah.
is
Yeah,
causing something
right,
small.
right.
But the relationship is an etiological one, is a historical
It's
one.
yeah.
Yeah, but
It's
you know,
etiological.
I actually have a question.
Uh-huh.
Just-
Okay. Okay.
Or I guess I have another commentary on something else.
Sure.
I think we need to stop thinking that the only kind of TM mechanism is expressed in that damn diagram.
Okay.
And I think Carl would agree with me that he's like tired of that. Talks, right?
But he had it in his own talk. He just had one more.
But I mean, you know, because one of the things is that there is an assumption, I think, that comes from looking at that. You know, when Carl
Yeah.
did that beautiful picture, which I love, I actually think that I can mouse the page.
Yeah.
There's a particular kind of phenomena that was trying to be explicated in the context of a particular project.
Right.
But clearly, the concept of mechanism has to talk about a set of causal relationships, which are not all like that. We have to talk about why one thing interacts with another. We need to talk about causal history. We need to talk about a lot of other things. And I think that the kind of thing that you're saying, oh, this isn't a mechanism, is because you're saying it's not that mouse in the maze.
Yeah.
And
Yeah.
but, you know, the thing that's interesting about that mouse in the maze is that what you're looking at is you're looking sort of in some sense synchronically and
Yeah.
saying,
Yeah.
here's something going
Yes.
on right now, and I want
Yeah.
to understand what's going on at the same time. Whereas in epigenetics,
Mhm.
you're looking
Right.
at the causal history.
Exactly.
Yes.
Exactly.
Yeah. Different kind of mechanism.
Yeah. Well, okay. Well, but again, it does not fit the strictures that I think of when I think of mechanisms. In particular, these nested hierarchies of mechanisms within mechanisms.
Right. But, you know, again.
And that's non-negotiable to the mechanistic picture.
Oh, it absolutely is. It absolutely is.
Well, it has not been in print.
The background idea to all of this was Wesley Salmon's view of
Yeah.
causal explanation,
Yeah,
which
yeah.
acknowledges both
Yeah.
etiological and
Yeah.
constitutive
Yeah.
explanations.
Yeah.
And the count of constitutive explanations is not thereby an assertion that those are the only kinds of explanations on offer.
Yeah.
That's it.
Yeah.
That's the simple point.
Oh, I'm sorry, Bob.
Yeah, good, exactly.
Okay, change of topic.
Sure.
I'm no expert on epigenetics.
Yeah.
But epigenetics is all over the place.
Yeah.
You might benefit from.
Can we get a mic so everybody can hear, Bob?
Oh, yeah, but you had me.
Yeah, where is it?
It's right next to you.
I want to make sure everybody can hear him.
Right where
Yeah, that's telling me to find it.
Sorry, my voice is
Here it is, right here. Here you go, Bob. Great. I want to make sure everybody can hear you.
Sorry, my voice is failing.
You bet.
Epigenetics is pretty common. It was surprising when it was discovered,
Mm-hmm.
but it shows up, for example, in turtles.
Yeah.
Where the same genotype can turn out to be male or female depending on the temperature of the sand in which they're raised or in which they hatch. It happens all over the place.
Yeah, you bet.
So there's no question that your description of this is, if this weren't absolutely real, something like it would be. And I think it is one level,
Mhm.
much as I understand the one level.
Mhm. Yeah.
I think the picture, the hadn't occurred to me till today, but the picture that Carl gives of interlevel with the two circles,
Mhm.
or more than two circles, is troubling because it, among other things, it makes me think about the disputes around Kim,
Mhm.
reductionism, which are
Mhm.
mistakes.
Mhm.
But because it does have this top-down and
Yeah.
bottom-up causation
Mhm.
story, I don't know why you need that. And I don't know why you couldn't simply say, for example,
Yeah.
decomposition and localization require two levels,
Mhm.
but decomposition and localization are only one way of revealing mechanisms.
Okay.
You might find it just by intervening.
Yeah. But that's very much the Silver Lantern Bickle picture, right?
Yeah.
That's very much what it is, right? I'll let you finish, but that idea right there, I think, is very much like what we were trying to discuss. The fact that everything looks, kind of going down to the level of molecules is because, well, that's where the interventions are in molecular and cellular cognition. We've got these cool tools that enable us to intervene at that level and track behavioral effects, and they're significant. And now we can even track system-wide effects. We can do molecular systems biology, as Alcina likes to say.
Well, yeah, I mean, you attach a methyl
Yeah.
compound to the
Yeah.
genes.
Yeah.
It
Yeah.
changes their expression.
Yeah, you bet. Yeah.
And it's inheritable.
Yeah, and here's these cool tools.
So that's pretty, but
Yeah.
I don't know why we need the circles.
Okay. But again, I mean, but again, and in some sense, what the Silver Lantern Bickle picture does, I suggest, is that it captures what is dead on right about mechanism. Okay, but it does it simply in a way that does not bring anything of these salmon-like pictures into the effect. It's just describing the way that the experiments are done in this branch of science that claims to find evidence for molecular mechanisms of a cognitive function. That's the way these conclusions are going. And all it tries to do is say, okay, how in the world do you go about doing experiments where you would draw that conclusion? How do you design them? And it's simply attempt to be purely descriptive without any kind of assumptions about what causes are, or about, whether we have these nested hierarchies of mechanisms or what levels are, anything like that. Just here's the way the science is done in this field of neuroscience. And I think so. Everything that comes out of that picture, everything that's right about mechanism comes out of that picture, without having to sort of think you've got some kind of grand ontological structures that you're articulating, because we're just trying to describe experiments and the kinds of conclusions that this particular group of scientists draw from doing these kinds of experiments and getting these kinds of results.
But you also need and want some nested hierarchies of mechanisms in the other sense.
Yeah.
Right? That is, you may have something invested in maternal behavior. You also need to explain how various molecular things contribute to that.
Yeah.
But you'll characterize
Yes.
those mechanisms in
Yes.
slightly different ways.
Yeah.
There will still be a hierarchy of mechanisms.
Yeah.
It's just that as you move down the hierarchy, they do different work.
Good. Because here's what I think you do. Say if you just follow still the Landreth Bickle lines, okay? We've now established by these particular kinds of experiments, we've now established that maternal behavior causes those mechanisms of epigenetic gene expression in that particular gene. Okay. We've established that because we've done our correlation studies, we've done our negative interventions, we've done our positive interventions. Okay, now, so we've got that. Now, what's the next step that you do if you're Silva, Landreth, and Bickle? Okay, what are the mediating causes that actually connect up these two phenomena? And now we'll go do those experiments until we can get convergent 3 evidence for those when we sort of look for these mediating causes. Well, okay, we'll see what we find, okay? But the idea is that simply we can remain neutral on what counts as a cause, what counts as an effect. We don't have to put any strictures on that, but just simply see what in the world the scientists doing this research are doing to establish their conclusions, articulating that kind of framework of experiments, and how they go about integrating their studies, and just this purely descriptive enterprise about a particular practice of science. What's that?
The conclusions aren't causal.
I think they are. I think they talk causally. Yeah, they sure do.
I thought you thought we were avoiding causal.
Well, no. We're not. We're avoiding any kind of presupposition of what causes are.
But Salmon has an answer.
I know he does. Okay, but you don't have to buy it. And still you can capture everything by just simply studying the experimental designs and studying the results and studying the conclusions in these particular areas of science. You can tell the story about how the practices of this science work. Now, yeah, they talk causally, okay? So the part of what you're explicating is a story about causal conclusions. But you don't have any presupposition in the background about what those things might be, what those causes might be.
Yeah, I want to change the topic again.
Sure, yeah.
I'm all fired up by Jackie's talk.
Uh-huh.
So
Yeah.
I'm curious about the social constraints
Yeah,
that
good.
are driving us to do this kind of research.
Good.
And Jackie pointed out, if I remember right,
Yep.
was it $50 million
Yeah.
to get something to a trial, and then
Yeah.
$100 million
Yeah.
to get it from the trial, so for a drug or something like that?
When you get up that high,
Yeah, exactly.
One's $1 billion.
Exactly.
Oh, $1 billion to $1 billion.
Yeah.
All right.
Yeah.
Why
Good.
Not, why not like classes on parenting?
What's that? Okay, classes on.
Parenting. Why not research on
Yeah.
teaching
You-
parenting methods?
That's exactly what this
is
suggests.
Why are we directing our energy at learning the molecular mechanisms that underlie the effects of rats licking one another, rather than putting money to solve this problem into social programs or to research on the kinds of social programs that would address it directly?
Hey, talk with the funding agencies. I'm just here's a branch of very well-funded science. Very well-funded science.
That's granted. I'm asking why fund that?
Go talk to an NIH officer.
No,
don't ask me. I'm just trying to tell you how this science works.
So why not do research at the higher level to figure out how parents should interact with their kids?
I guess because drug companies have far too much influence over NIH. And drug companies aren't going to make much money about figuring out a way that parents could do better, and they've got far too much influence over NIH.
So,
sort of like what vaccine companies have over NIH, because a particular person was the head of the infectious disease. I didn't put that cartoon up there for nothing. Was the head of the infectious disease area who totally screwed up the response to AIDS by going and looking for vaccines instead of therapeutics. And guess what? He totally the treatment of COVID because he only funded vaccine research. And of course, what do we know? How do you take Paxlovid and you tend not to be too bad unless you're really sick.
Okay. Question here.
It kind of question you kind of playing off of what
Yeah.
Carl just said.
Good.
I'm really glad that you brought that up because that was a question I wanted to
Good.
ask, but I wasn't sure. if
No,
it was
good.
going to be
Great.
Appropriate.
Yeah,
but
yeah.
as
I'm
you
not sure it
mentioned-
was.
It was.
Yeah, but as you mentioned, there are studies that have shown that child abuse victims have methylation and
Yep.
these glucocorticoid
Mm-hmm.
receptors.
Yep.
And so I was wondering how you think that would translate to mental health care and to things like parenting.
Yeah.
And I think kind of this isn't an answer to your question. It's more so a response. I do think that studies like this are important because oftentimes what the public wants and what parents want are good reasons on why they should take these classes and why they should care about learning more about how to be better parents. And if we can say, hey, look, the way that you parent impacts brain function, neuronal growth, then you have maybe better reasons in the public eye to care more about parenting. And so I personally find stuff like this to be really valuable to fund. However, I also agree that translating it to the social realm and to something that parents care about is really important.
Oh, yeah, it's really important and it's really difficult. All right, but the evidence, the not just the neuroepigenetic evidence, but the epigenetic evidence really suggests that that particular set of external causes is really crucial, not just as a special input into this already existing mechanism, but literally a part of the causal network that's literally building the gene expression system. That's going to persist into adulthood. Yeah, I think that's what's really cool about this research. I agree completely.
Any final quick questions? All right, well, if not, let's thank our speakers.
Thanks, man. Thank all y'all.